Hello, I'm Professor Sims, and in this video I'll discuss the modern understandings of inheritance. This is the third in the series of 10 lessons held as part of my general genetics course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include discussing Sutton's chromosomal theory of inheritance, describing genetic linkage and how karyograms are created, explaining the process of homologous recombination, calculating the distances between genes using a 3-point test cross, examining how nondisjunction leads to disorders in chromosome number, comparing types of disorders that Ploidy causes, and describing how errors in structure occur via inversions and translocations. So we're going to be talking about mostly Sutton's chromosomal theory of inheritance, but first we got to make sure we go back and understand Mendel's chromosomal theory of inheritance. It was supported by the following observations. Okay, so you have homologous chromosome pairs migrate independently during meiosis. Sorting of chromosomes from each pair into pre-gametes appears to be a random process. Each parent produces gametes with half the chromosomal complement. The sperm and egg, despite their differences in size and shape, have the same number of chromosomes, which suggests equal genetic contribution from each parent. And the gametes combine during fertilization, which results in offspring that have the same chromosome number as their parent. With this, Gregor Mendel laid the foundation for modern genetic inheritance, but his early work was completed in the 1840s before the discovery of chromosomes under the microscope. Further, his work was met with resistance in the scientific community, mostly because there are examples in nature of blended inheritance, and because individuals have far more independently segregating traits than they had chromosomes. Advancements in microscopic techniques allowed for cell biologists to stain and visualize subcellular structures, and eventually they were able to witness the actions of chromosomes during cell division and meiosis. But before that, in 1902, we had Theodore Boveri, and he was able to determine that inheritance of chromosomes was essential for the development of sea urchin offspring. That same year, Walter Sutton was able to witness chromosome separation during meiosis, and this supported Mendel's theory of inheritance. Then we have Thomas Morgan further supported the theory through his extensive fruit fly crosses, and this provided experimental evidence in favor of the chromosomal theory of inheritance because it revealed a connection between a segregating trait and the X chromosome, disrupting Mendel's predicted outcomes. So this led to scientists realizing that genes located on the same chromosome can sometimes become unlinked during meiosis. In 1913, Eleanor Carruthers was the first person that was able to provide physical evidence. So up until now, it was just kind of mathematical, observational. She had physical evidence because she was able to observe independent chromosome assortment under the microscope during meiosis. One of Morgan's fly experiments involving body color and wing shape demonstrated the occurrence of recombination events leading to non-parental phenotype. So this is how he was able to reveal the segregating traits, right, without the use of a microscope. Now, please have a look through this figure. It's figure 13.3 in the book, because I know it's very difficult to see here, but I'm going to briefly go over this. Okay, so these experiments show unlinked and linked gene inheritance patterns. In this top section A, two genes are on different chromosomes, so independent assortment occurs, and the offspring have an equal chance of inheriting the parental type or the non-parental type. On the left here in section B, this is demonstrating where two genes, they're very close together on the same chromosome, so there's no crossing over. The genes are inherited together, and all the offspring have the parental type. Then over here, section C on the right, this is demonstrating two genes that are far apart on the chromosome, so crossing over does occur during meiosis, and the recombination frequency will be the same as if the genes were on separate chromosomes. And then lastly, D see down here is showing the actual recombination frequency of fruit fly wing length and body color that Thomas Morgan observed, and it was 17%. And a crossover frequency between 0% and 50% indicates that genes are on the same chromosome, and that crossover sometimes occurs. Let's go a little bit deeper into homologous recombination. In 1909, Franz Janssen observed chiasmata, which are points where chromatids make contact and exchange segments during the first meiotic division. This suggests that chromosomes physically exchange segments and alleles become unlinked. Now, synapsis, that's the pairing and interaction of homologous chromosomes, and it leads to recombination. We saw this when we were discussing meiosis. And this is how new allele combinations and new non-parental types are formed. So to illustrate this quickly, let's just consider an example of a heterozygous individual with dominant maternal alleles and recessive paternal alleles. So mom's capital A, capital B, dad is lowercase a, lowercase b, okay? So if the genes are linked, we would expect a 1-to-1 ratio of capital A, capital B, and lowercase a, lowercase b gametes. However, if the genes are not linked, the individual should produce capital A, capital B, capital A, little a, capital B, and little a, little b gametes in equal frequencies instead. So what he did was Morgan discovered that both parental and non-parental cases occurred when test crossing individuals with a homozygous recessive parent. And what this indicates is that while linkage is the majority outcome, a significant portion of offspring resulted from recombination. This figure here illustrates the chiasmata that Janssen observed. And notice these are the same synapses that we saw when we were discussing meiosis in lesson 1. Let's watch this short video that further discusses the chromosomal theory and recombination.
Mendel's work was far beyond excellence. However, it wasn't appreciated right from the time it was put forth before the scientific society. Several reasons led to the demotivating rejection of his efforts. One of the major reasons included his concept of non-blending inheritance. According to Mendel, genes, or factors to be precise, do not blend, and that the phenotype of the offsprings is similar to only one parent. It cannot be an intermediate between the two parents. However, the variations seen in organisms in general could not be explained by this concept of non-blending of characters. Secondly, Mendel introduced mathematical calculations to explain biological processes. He presented results in the form of ratios. At that time, mathematical forms of biological results were not easily accepted. Hence, his work was largely ignored. Lastly, when Mendel said that there are factors which are passed from one generation to the other, he had no physical proof of those units. In the sense, there was no evidence of the physical form of the factors that Mendel could present. So the proposition that discrete units called factors exist was like a hypothetical theory. Mendel had no proper evidence that such units actually exist. Due to all these reasons, Mendelian genetics remained dormant for several years. After being almost rejected in the mid-1860s, Mendel's work remained unnoticed until the beginning of the 20th century. So what exactly happened in the early 1900s? Who shined a light on the remarkable contribution of Gregor Mendel? Let's find that out in the next part. Microscopes ease the lives of scientists. However, they are not new inventions. Microscopes were invented several years ago. Beginning with simple lenses used to zoom into the desired structures, today we have electron microscopes which can give us minute details of the smallest possible structure. But the microscopes in the 19th century were not that advanced. So minute observations of the living cells and the cellular components were not really possible. In the beginning of the 20th century, the advancements in microscopy and related techniques helped in acute observations of the cell division processes. As a result, cytologists could now figure out structures that duplicate and divide during cell division. Which structures could these be? That's right. Scientists were now able to identify and spot chromosomes inside the nucleus. We now know what chromosomes are and how they look, but it wasn't known back then. So the newly found structures inside the cell grabbed a lot of attention. Also, by this time, due to the understanding of the chromosomal structures, processes like mitosis and meiosis were well understood. During the same time, Mendel's long-ignored legendary work started gaining importance as many scientists started re-evaluating the results. There were two scientists from different parts of the globe who simultaneously put forth a few hypotheses. These hypotheses became a theory later, which is what we are going to study now. Walter Sutton, an American geneticist and physician, was working on grasshoppers. He found that chromosomes occur in pairs and during meiosis separate from one another in the gametes formed. He put forth the assumption that a pair of chromosomes contains one chromosome from the mother and one from the father. The separation of chromosomes during meiosis may be the explanation of the Mendelian laws of inheritance. This is what Sutton hypothesized. Around the same time, a German biologist named Theodor Boveri was working on sea urchins. He found that in order to have a proper embryonic development, all the chromosomal pairs have to be present in the embryo. Both these works later were combined to give us a single theory, which came to be known as the Boveri-Sutton theory of inheritance. Now, it's also referred to as the chromosomal theory of inheritance. The theory, in simple words, stated that individual genes are found at specific locations on a particular chromosome. Also, the pattern of behavior of the chromosomes during meiosis explains us the Mendelian laws as to why and how the genes are inherited. The theory, and thus the laws of genetics, can be explained well with the help of exactly 3 points or observations. The first point said that chromosomes come in pairs. These are now called homologous pairs by us. One chromosome from the pair comes from the mother, while the other from the father. Isn't this what Mendel had hypothesized about genes too? He said that factors, now called genes, come in pairs. One factor of the pair is from the mother and the other from the father. The next point stated that the chromosomes from a single pair separate from each other during the process of meiosis. That means one chromosome will be passed to the respective gamete. This is just the simplest explanation of Mendel's law of segregation. In the law, Mendel emphasized on the separation of factors, that is alleles, into gametes. The last observation of the theory states that different chromosomes assort into gametes independent of each other during meiosis. This is what even Mendel had said in his law of independent assortment. The alleles of the different genes assort independent of each other during gamete formation. This is how the chromosomal theory of inheritance helps us understand that the genes are present on chromosomes, and also that they are the units of heredity. This is how the theory supported Mendel's work and was useful in proving the laws of genetics put forth by Mendel. However, now the question is, how was this theory accepted? What results were presented to prove this theory right and acceptable? A few years later, another legendary scientist named Thomas Hunt Morgan carried out a few experiments on the little fruit fly Drosophila melanogaster. His experiments confirmed Sutton and Bovary's theory further.
Now let's move on to genetic linkage and mapping. So in 1913, Alfred Sturtevant created the first chromosome map, and this map represented gene order and relative distance on a chromosome based on recombination frequency. So he assumed that genes were serially ordered on a chromosome and that recombination occurs with equal likelihood along the chromosome's length. He postulated that alleles that are far apart on a chromosome were more likely to recombine during meiosis due to a larger recombination region. The genetic map was divided into map units, or centimorgans, cm, where a 0.01 recombination frequency corresponds to 1 cm. Sturtevant's map allowed the calculation of distances between genes on the same chromosome. So perfectly unlinked genes will have a recombination frequency of 0, while genes with a frequency of 0.5 are totally linked, 100% linked, or they're on different chromosomes. Figure 13.4 shows that by using recombination frequency to predict genetic distance, we can infer the relative gene order on chromosome 2. So the values here represent map distances in centimorgans, which correspond to recombination frequencies in percent. So the genes for body color and wing size were 65.5 minus 48.5 equals 17 centimorgans apart. And that indicates that maternal and paternal alleles for the genes recombine in 17% of the offspring on average. In 1931, Barbara McClintock and Harriet Creighton demonstrated that there's crossover in homologous chromosomes in corn plants. And then weeks later, Kurt Stern demonstrated microscopically that homologous recombination happens in Drosophila fruit flies. Stern observed several X-linked phenotypes that were associated with a structurally unusual and dissimilar X chromosome pair, in which one X was missing a small terminal segment and the other X was fused to a piece of the Y chromosome. By crossing flies, observing their offspring, and then visualizing the offspring's chromosomes, Stern was able to demonstrate that every time the offspring allele combination deviated from either of the parental combinations, there was a corresponding exchange of an X chromosome segment. Using mutant flies with structurally distinct X chromosomes was the key to observing the products of recombination because DNA sequencing and other molecular tools were not yet available. Recently, though, geneticists did the mapping. They did the math for the 7 traits that Mendel was studying. We talked about this in the last lesson. So the 7 traits that Mendel was studying in the pea plant genome were unlinked. So it was totally confirmed that these genes are either on separate chromosomes or they're far enough apart on the same chromosome to be statistically unlinked. And some people speculate that Mendel was either really lucky to select unlinked genes, or did he ignore data that suggested linkage? But nevertheless, Mendel consistently observed independent assortment because he was looking at unlinked genes. Chromosome isolation and microscopic observation forms the basis of cytogenetics, and it's the primary method by which clinicians detect chromosomal abnormalities in humans. Chromosomal abnormalities during meiosis can lead to inherited disorders. And these abnormalities can be classified into 2 categories. The first one is changes in chromosome number, and the second one is structural rearrangements, and we're going to talk about both of those. Cytogenetic analysis involves isolating and observing chromosomes under a microscope, and it's helped greatly by using a karyotype, or also known as a karyogram. What this does is it provides a visual representation of an individual's chromosomes, including their length, their banding pattern, and the position of the centromeres. Chromosome number disorders are easily identified using a karyogram, and they usually result from duplications or losses of an entire chromosome, or a change in the number of sets of chromosomes. We'll go into this in some more detail in just a bit, but I did want to show you figure 13.5. This is a karyotype, or also known as a karyogram, of a female human. So you can see the homologous pairs of chromosomes. They are the same length, the same size, they have the same centromere positions, and they have very similar banding patterns. Okay. You know that this is a female because she has 2 of the X chromosome. If this was a male, it would be an X and then a smaller Y chromosome here. I'd like you to see this short clip that describes how karyotypes or karyograms are actually made in the lab.
A karyotype is an individual's set of chromosomes. The term also refers to a laboratory-produced image of a person's chromosomes, arranged according to well-defined criteria. The first step in performing a karyotype is to collect a sample. A blood sample containing red blood cells, white blood cells, platelets, and plasma. The second step is separating the cells. A karyotype will be done on the white blood cells since they actively divide, and contain a nucleus where the chromosomes are located. The third step is growing cells. In order to have enough cells to analyze, the dividing cells are grown in a special medium at 37 degrees Celsius. This process of culturing can take 3 to 4 days. The fourth step is blocking cell division. The cells are treated with a chemical substance called colchicine, which blocks cell division in metaphase where the chromosomes are in their most compact form, and consequently, they are easily observed under a microscope. The fifth step is releasing the chromosomes from their cells. In order to see these compact chromosomes under a microscope, the chromosomes have to be out of the white blood cells. This is done by treating the white blood cells with a salt solution that causes them to burst. The sixth step is staining the chromosomes. Chromosomes are naturally colorless. In order to tell one chromosome from another, a special dye called Giemsa dye is applied to the slide. The final step is cutting and arrangement. Once chromosomes are stained, the slide is put under the microscope for analysis. A picture is then taken of the chromosomes. By the end of the analysis, the total number of chromosomes will be determined and the chromosomes arranged.
So like I said a minute ago, the chromosome number abnormalities are most easily identified by looking at a karyogram, and it's going to involve duplication of a chromosome or loss of an entire chromosome, and it actually changes the number of chromosome sets that you have in the nucleus. And the main cause of this is nondisjunction. Nondisjunction occurs when homologous chromosomes or sister chromatids fail to separate during meiosis. Some causes of nondisjunction include misaligned synapses, an incomplete synapsis, or a dysfunction in the spindle apparatus. And it can happen during meiosis 1 or meiosis 2. If it happens during meiosis 1, then you have 2 gametes that are missing a chromosome and 2 gametes that have an extra chromosome. Conversely, if nondisjunction happens during meiosis 2, then 1 gamete will have a missing chromosome, 2 gametes will have the correct number of chromosomes, and then 1 gamete will have an extra chromosome. And of course, this difference is due to the difference in the 2 cell division cycles, right? So in one cycle, you have chromosomes being pulled apart. In the other cycle, you have chromatids being pulled apart. Figure 13.6 shows how nondisjunction occurs in either meiosis 1 or meiosis 2. So the error is occurring either when a pair of chromosomes fail to separate or when a pair of sister chromatids fail to separate. And we see here in the offspring, we have 2 gametes. And if it's happening during meiosis 1, we have 2 gametes that are missing a chromosome and 2 that have an extra one. And if it's happening in meiosis 2, then we have these 2 gametes have the normal number, But these ones, this guy's missing one and this one has an extra one. Let's talk about ploidy and some of the terminology around it. So we have euploid, aneuploid, and polyploid, and these are all terms that are used to describe different chromosome conditions. Okay. Euploid refers to individuals who have the correct and balanced number of chromosomes for their species. In humans, euploidy corresponds to having 22 pairs of autosomes, which are the non-sex chromosomes, and 1 pair of sex chromosomes, and the total is 46 chromosomes in each cell nucleus. That's euploid. Aneuploid individuals have an abnormal number of chromosomes due to the loss or gain of 1 or more chromosomes. Aneuploidy can occur during cell division, when chromosomes fail to separate, or like we just saw, when sister chromatids fail to separate properly. Now, I've got a couple examples here of aneuploid associated disorders in humans. Monosomy is a type of aneuploidy where 1 chromosome is missing. So the result is having 45 chromosomes in the cell nuclei instead of the normal 46. Pregnancies with an autosomal monosomy usually end in embryonic death. Now, monosomy of the X chromosome, this is the exception. This is the only non-lethal monosomy. It's called Turner syndrome, and it's a condition that occurs as a result of a missing or sometimes partially missing X chromosome. It causes a variety of medical and developmental problems, including short height, failure of the ovaries to develop, and sometimes even heart defects. Trisomy is another type of aneuploidy where there's an extra chromosome. An example of trisomy in humans is trisomy 21, which is also known as Down syndrome. Down syndrome is a condition in which a person has 3 copies of chromosome 21 instead of the normal 2 copies. This extra copy changes how a baby's body and brain develops, and it causes mild to moderate intellectual and physical disabilities that last a lifetime. Because the likelihood that an egg will contain an extra copy of chromosome 21 increases significantly as a woman ages, older women are more likely than younger women to give birth to an infant with Down syndrome. Figure 13.7 is showing the incidence of having a fetus with trisomy 21 and how it increases dramatically after the age of 40. Polyploid organisms have multiple sets of chromosomes beyond the normal diploid number. So remember, the normal diploid number in humans is 46. Some examples of polyploidy are triploid organisms. They have 3 sets of chromosomes, so they're 3N. Then you have tetraploid, those are 4N, so on and so on and so on. Polyploidy often arises due to errors in cell division, just like we've seen before, and it leads to the doubling or the addition of a chromosome. And what you end up with is an organism that has too many sets of chromosomes. Now, this is actually really common in plants because in plants, polyploidy tends to make the plant larger and more robust, and it's more adaptable, right? So figure 13.8 here is showing triploid orange daylily. So not only is it larger, it's more robust, but it also has more petals and more stamen, and it can reproduce better than if it was diploid. Now, this is not a common occurrence in animals because polyploidy in animals often results in early embryonic loss. And even if the animal survives, it's usually going to be sterile and doesn't live often to reproductive age anyway. Variations in the number of sex chromosomes have relatively mild effects as compared to those we've seen for autosomes. So X inactivation, this is a molecular process in female mammal embryos, where 1 X chromosome in each cell becomes dormant and forms what is known as a bar body. This can occur randomly, but females can compensate for it sometimes with their other X chromosome. But if there is no X chromosome present at all, the offspring are not going to develop in utero. Abnormal numbers of X chromosome lead to X inactivation in each cell, resulting in mild intellectual and physical disorders, disabilities, and often sterility. X chromosomal triplo X, they're XXX individuals. They are phenotypically female, and they may have developmental delays and reduced fertility. Now, XXY individuals, this is called Klinefelter syndrome, and these individuals are phenotypically male, but they have small testes, they have enlarged breasts, and they usually have reduced body hair. More complex types of Klinefelter syndrome may have as many as 5 X chromosomes, with multiple bar bodies in each cell nucleus. Duplications and deletions are another type of chromosomal abnormality that can lead to offspring with abnormalities. Duplicated chromosomal segments may join existing chromosomes, or they might exist independently in the nucleus. Cry-Duchat syndrome is a notable example where a deletion of most of the small arm of chromosome 5 causes nervous system abnormalities and distinct physical features. Infants with this syndrome emit a high-pitched cry, and that's how it got its name, Cry-Duchat. I'm probably saying that wrong. Cry-Duchat syndrome is a rare genetic disorder, and it's caused, again, by missing pieces on chromosome number 5. In addition to the high-pitched cries that you see in infants, other symptoms include a small head and a flattened bridge of the nose. Figure 1310 is showing an individual with this syndrome at ages 2, 4, 9, and 12 years of age. Structural rearrangements of chromosomes, such as inversions and translocations, have been extensively studied. So during meiosis, rearranged chromosomes can pair up with their former homologs to maintain proper gene alignment. However, if the gene orientations on the homologs are incorrect, recombination events can result in the loss or gain of genes, leading to aneuploid gametes. So chromosome inversions involve the detachment, 180-degree rotation, and reinsertion of a chromosomal segment. Inversions can occur naturally due to the mechanical shear or the action of transposable elements. Unless they disrupt gene sequences, inversions usually have milder effects than aneuploid errors. However, changes in gene orientation can affect gene expression, regulation, potentially causing abnormal levels of gene products. Pericentric inversions involve the centromere, while paracentric inversions occur outside of the centromere. Pericentric inversions can alter the relative lengths of the chromosome arms, making them easily identifiable. In inversion heterozygotes, one homologous chromosome undergoes an inversion, while the other remains unchanged. During meiosis, one homolog forms a loop, and the other molds itself around the loop to maintain synapsis. Although this ensures correct gene alignment, it can result in stretching and imprecise synapsis regions. Figure 1311 helps to illustrate pericentric inversion versus paracentric inversion. Pericentric inversion can change the length of the chromosome arms above and below the centromere. And remember that paracentric inversions do not change the length of the chromosome because they are not happening around the centromere. So just imagine if you have, say, E is twice as long as F, and C is twice as long as D, okay? If you were to invert D and C, then what will happen is this arm of the chromosome will end up being shorter than this arm. Whereas if you were to switch F and E, the length of the arms is unchanged. So that's the main difference between these two types of inversion. Figure 1312 shows the process of inversion pairing. Inversion pairing is that process we were talking about before, where it ensures proper alignment and exchange of genetic material during meiosis in organisms that have a chromosomal inversion. So the process involves the formation of a loop in one of the homologous chromosomes, and this helps it to achieve accurate pairing and successful crossover during the synapsis. Translocations occur when a segment of one chromosome dissociates and attaches to a different non-homologous chromosome. The effects of translocations depend on how gene positions are altered in relation to regulatory sequences. So certain translocations have been associated with cancer and schizophrenia. Reciprocal translocations involve the exchange of chromosome segments between non-homologous chromosomes without either chromosome gaining or losing genetic information. Figure 1313 illustrates the process called reciprocal translocation. It is important to note that this process can lead to new combinations of genes, but It doesn't alter the overall genetic content of the chromosomes that are involved. Before we finish up today, I want to show you a clip from this video that helps to summarize several of the topics that we covered in this lesson.
By now we know what a karyotype is. Can you try to give me some conclusions of it? You may say that it looks perfectly normal. But let me spot a flaw for you. Do you notice this set? This is the 21st chromosome set. Instead of having a normal pair of chromosomes, here we find a triplet. Now you may say that it may be good for the individual as it has an extra chromosome. But that's not the case. Any change in the number of chromosomes creates a lot of defects in the normal functioning of the body. On this note, let's understand chromosomal mutations. As the name says, chromosomal mutation means the mutation of chromosomes. And how exactly does it get mutated? It can happen in two ways. One way is where the structure of the chromosomes gets disrupted. The other is where the number of chromosomes are different than the usual in the organism. Let's have a look at each type. This is the structure of a typical chromosome. Now imagine this small part gets physically removed. This would result the chromosome in getting smaller as its segment is lost. This also means that if the segment had some important genes, then they will also get lost, which can result in severe abnormalities. Such type of a mutation is called deletion. This is where the part of the chromosome gets deleted. Now imagine this part of the chromosome gets duplicated. As a result, the chromosome's length increases. Needless to say, this will also result in some or the other abnormality in the individual. This type of mutation is called duplication. Now comes the next type. These are two different gene segments. Imagine they get flipped. Thus, the chromosome will appear like this. This is called inversion. Do you think this type of mutation is harmful? It may or may not be harmful depending on which genes get flipped. Now let's take one more chromosome. Imagine a part of this chromosome gets removed and gets inserted in this chromosome. What will be the result? That's correct. Both the chromosomes will change drastically. This type of mutation is called as insertion. And now comes the last type. Instead of a segment getting inserted to another chromosome, let's imagine a situation where a segment from each chromosome gets interexchanged. So this segment will get into this chromosome and vice versa. This kind of mutation also results in abnormalities as both the chromosomes get altered completely. Any idea what this type will be named as? Well, this type of mutation is called translocation. All these mutations resulting in the disruption of normal chromosomal structures are broadly capped under a single heading. They are categorized as chromosomal aberrations. Now, this was about the mutations that involve changes in the structure of the chromosomes. What about the mutations that involve a change in the number of chromosomes? Let's understand that in the next part. This is a normal chromosome, while this is a disrupted one. When the structure of a chromosome gets altered, it's called chromosomal aberration. We have seen this. Now let's move on to the second type. It's the mutation that involves a change in the number of chromosomes. It's called as aneuploidy. In literal terms, aneuploidy means the presence of an abnormal number of chromosomes in a cell. So the karyotype of a person with trisomy 21 is an example of aneuploidy. The presence of one additional chromosome in the 21st set gives rise to a total of 47 chromosomes in a person. So instead of a normal 46 chromosomes, this person has an additional chromosome. To be precise, an additional autosome. This case of aneuploidy is called trisomy 21. This is because there are 3 chromosomes instead of 2 in the 21st set. The problem of trisomy 21 leads to a condition called Down syndrome. We will get into the details of these disorders in our higher grades. But for now, let's focus on understanding the chromosomal mutations. This aneuploidy involved an autosome. Now tell me, what if the number of sex chromosome alters? So instead of XY, what if an individual has XXY? Is that even possible in the first place? Well, yes. There can be individuals with an additional X chromosome. This condition is called Klinefelter syndrome. Such individuals exhibit both masculine as well as feminine characteristics. Now, in the case of females, imagine that instead of addition, there is a deletion of an X chromosome. This would mean XO. So this individual will have only one X chromosome. This condition is called Turner syndrome. There are a few developmental abnormalities in such scenarios. These individuals have a total of 45 chromosomes only.
That's going to conclude today's material for Lesson 3. Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics and leave your questions for me in the comments below.